Okay guys, I want to give you a basic overview of everything we went over throughout the chapter with cell theory, prokaryotes, eukaryotes, animal cells, plant cells, all that stuff. But just to start off, cell theory, there was three parts of it. The first part said that all living things are made of cells, and that was it. Nothing, you guys learned that in probably 4th grade. The second part said that cells are the basic unit of structure and function in living things. What that means is essentially, guys, cells are going to make up everything, and they are going to not only make things up, but start to build those things up into bigger things by combining more cells together. And then the last part says that cells reproduce. Simple as that. Meaning that one cell can actually give rise to more cells, and that's how we get more cells. All right, but just to start off, guys, these are all our characters. I know they're emojis. They're not really emojis. Well, they weren't emojis in real life. I don't think emojis were created back then. So it all started about the 1600s. The first guy, his name was Robert Hooke. He was experimenting with microscopes, and he started to look at tree bark under the microscopes, and he noticed they were all made up of little blocks, almost cells. And tree bark, guys, is obviously a plant cell, and we know plant cells have the square shape to them. So that's why he thought, okay, they're square, they look like little dorms of monks at the time. Again, guys, that was like 400 years ago, but that's why he called them cells, is because the monks called their dormitories cells. All right, so Robert Hooke, he was our first guy. He literally discovered cells, was the first guy to discover them, again, using his microscopes. Around that same time, there was a guy named Anton von Leeuwenhoek, and he was also using microscopes. And he found that every single bacteria that he was looking at under his microscope, all these bacteria were made of cells as well. So we had Hooke, he said cells exist. He was the first one to discover them. Leeuwenhoek, shortly after, said, okay, bacteria is made of cells. 200 years go by, so now we're in the 1800s, all right? It's almost like going back to the atom, guys. The atom was originally discovered by like Democritus, like in BC times, a long time ago. We didn't start like researching the atom actually and finding things until the 1800s. So, many years go by with these things, and we slowly start to group them together and add these theories together to come up with a bigger theory. In this case, it was our cell theory. But in the 1800s, we had these two guys, Schleiden and Schwann. Schleiden, what he did was he was looking at a bunch of animal cells under plant cells under a microscope, and Schleiden concluded that all plants are made of cells. Schwann, a swan is an animal, okay? Schwann was looking at animal cells under a microscope, and Schwann concluded that all animals are made of cells. So again, guys, we had Hooke, cells exist. Leeuwenhoek, all bacteria is made up of cells. Schleiden, all plants are made of cells. Schwann, all animals are made of cells. And during this time of the 1800s, guys, these three guys came to find their conclusions as well. The first guy, his name was Brown. He discovered that there was, in eukaryotic cells, there was this big globule in the middle of the cell, and he termed it the nucleus. So Brown discovered the nucleus. Virchow, what he did was, when he was experimenting, he noticed that the cells were becoming more plentiful at certain times. So he concluded that cells reproduce, all right? That was Rudolf Virchow. Last guy, his name was Felix Dujardin. What Dujardin did was he noticed there was essentially a substance in the middle of the cell, the cytoplasm or the cytosol, and he concluded that the cells have a fluid-like structure inside of them called the cytosol, all right? So that's everything for the cell theory, guys, and for all of our characters who contributed to it. So we will get rid of all of these guys. Now, to start talking about the cells, we had two different main types of cells. We had prokaryotic cells and we had eukaryotic cells, all right? So just to give you a rough overview of them, our prokaryotic cell, there we go, all right? Very basic. It has a cell wall on the outside. On the inside of our cell wall, it also has a cell membrane. This whip-like structure in the back is called the flagellum. It helps our prokaryotic cell move. All right, I guess I should label this for you guys. This is a prokaryote. Okay. Along with this, guys, our cell wall, our cell membrane, it's going to have some ribosomes throughout. Okay. They're going to be free ribosomes. They're not going to be connected to anything. They'll be kind of close to the cell membrane toward the outside. But that's pretty much all it has with respect to organelles like the eukaryotes do. And then lastly, it's going to have DNA in the middle. Now, we talked about the DNA of prokaryotic cells. It's a little bit different than the DNA of eukaryotic cells. Its DNA, it only has one of them, and if we were to unfold it, I know I did this in class with you guys, I got all the pipe cleaners and I had it all folded up, and then when we untangle all that, like I did with the pipe cleaners, it's going to create one circle, okay? So it only has one DNA or one chromosome, and in the end, that chromosome is going to be circular if we were to unravel it. And that's it with the prokaryotic cell, guys. Very simple, all right? If we're going to go with the eukaryotic cells, there's going to be two of them. There's going to be a plant cell and an animal cell. I'll start off with the animal cell. Okay. All right. So in your animal cell, it's going to have, I'll label it in pink like I did with the prokaryotic cell. It's going to have a cell membrane. Okay. Now the difference between prokaryotic cells and eukaryotic cells, the prokaryotic cell has DNA and it's pretty much floating throughout the cytoplasm, all right? So for eukaryotic cells, they're not going to have their DNA just going all throughout the cell. It's going to have a nucleus, and that nucleus is going to house, the DNA, all right? So there is your DNA, all right? There's also in the nucleus, which I'm not going to draw, is going to be a nucleolus in the middle. It's a very dense region, and its main goal is going to be to make ribosomes, all right? Now, as we're coming out of our nucleus, okay, all right. This one is going to be the rough ER, and in order for it to be the rough ER, you've got to put some ribosomes on it, okay? So there's your ribosomes on the rough ER. They're bound to it. Just like all ribosomes, their function is going to be to make proteins, all right? Now the difference between these ribosomes and the ribosomes over here, which we call free ribosomes, are these ribosomes are going to be making proteins that are going to be exported out of the cell, and they're going to go to other cells for those cells, whatever they need the protein for, all right? So again, this is the rough ER. Okay. Now, I'm just going to go through the pathway of a protein, guys, so I don't get this all congested with a bunch of organelles. I think this will be the best way to do it. All right. So our next guy here, he's going to be our Golgi. Okay. So what's going to happen here, guys, is that we are going to get the instructions from DNA. That DNA is going to be converted into RNA, and that's going to happen in the nucleus. After that, that RNA is going to leave the nucleus. It's going to go to one of these ribosomes, and that ribosome is going to convert the RNA into a protein. All right? So that protein is going to travel through the rough ER. It's going to get folded up a little bit, and then it's going to get exported out of the rough ER. All right? Now, we talked about these guys here, and that's going to house our protein, but he is called a vesicle, and it's essentially the car of the cell. That vesicle is going to hold the newly formed protein, and he's going to transport the protein to the Golgi. That vesicle is going to drop the Golgi, that vesicle is going to drop the protein off the Golgi. Protein's going to go through the Golgi, and it's going to get its tag for its final destination. So essentially what the Golgi does, guys, is he makes sure that that protein, again, it's going to get shipped out of the cell, is going to go to where it needs to go outside of the cell, all right? Essentially, it's Amazon, all right? If you order something from Amazon, you're going to have an address on your package, and that makes sure that UPS knows, okay, I know that this package has to go to this address. That's what the Golgi does. So that way, we're just not taking the protein, throwing it out of the cell. We know that it has to get somewhere, all right? Protein's going to come out on the other side, and then what we're going to do is we are going to export it out through the cell membrane. So again, guys, pathway of a protein, we start in the nucleus, all right? The DNA gets converted to RNA. We leave the nucleus, go to the rough ER. The ribosomes pick that RNA up. They convert that RNA into a protein. The protein leaves the rough ER through a vesicle, goes to the Golgi. The Golgi gives it where it's going to be shipped to. So essentially, guys, it's the packing. You know, like the packing label that they put on. That's what the Golgi does. It then gets once again put into a vesicle and shipped out through the cell membrane. All right. So that's all your major organelles that deal with your pathway of a protein. All right. So next, find a different color here. Over here, okay, this guy is going to be our smooth ER. Now the difference between the smooth ER and the rough ER is the smooth ER is not going to have any proteins on it. Okay, I'm sorry, ribosomes on it. So it's not going to be making proteins. Instead, our smooth ER is going to make our lipids, our fats. All right, it's going to help make the phospholipids for the cell membrane and all that kind of stuff. Okay, so for the smooth ER, guys, another thing it does is it detoxifies the cell. Essentially, like they're real plentiful in the liver cells because your liver helps to detoxify. All right. So again, rough ER has ribosomes on it, makes proteins. Smooth ER, no ribosomes on it, and it makes lipids. Okay. Other little organelles that we have in here, we're going to have some small, but they're still going to be there. Small vacuole. It's going to store water, store food, stuff we need. All right. We're also going to have a lysosome. And there could be more than one. I'm just making one to save space. Okay. There's your lysosome. What the lysosome is going to do, guys, is it's going to ingest old organelles, old cells, break them apart. Okay, so it'd be a bunch of hydrolysis reactions from what we learned from the macromolecule chapter. Break them apart, and those products, after the lysosome breaks it down, those products will be reused by the cell for whatever it needs, maybe to make a whole new organelle. All right, and then we're also going to have, and there will probably be more than one of these guys. I'm only drawing one. Okay, that is our mitochondria. And the mitochondria's goal is going to be to make 3 letters, ATP, which is our energy. Okay. You guys are also going to have a cytoskeleton. I'm not going to draw the cytoskeleton because if I draw it, it's going to be a bunch of lines going through the whole thing. And I don't want to completely cluster this picture up. But if you guys remember for the microtubules of the cytoskeleton, the microtubules are made out of the protein tubulin, and then the microfilaments are made out of the protein actin. All right. So those are your 2 parts of the cytoskeleton. And again, guys, they'd be, all throughout the whole thing. All right. The last thing that is what holds all of these organelles in place, which I didn't really go over yet. I talked about a little bit with prokaryotes, but there's also going to be cytoplasm or cytosol, the liquid portion. There's going to be that throughout, and it keeps the organelles in place. Okay. So if we're looking at similarities between the eukaryotic animal cell and the prokaryotic, so there's 4 similarities between prokaryotes and eukaryotes. All right. 1, they're all going to have DNA. So that's your first similarity. Your second similarity is going to be they're all going to have a cell membrane. Okay, so they're all going to have DNA, they're all going to have a cell membrane, okay, DNA, cell membrane. They're also all going to have ribosomes, so they can all make proteins, all right? They're free on here. I know on here I bound them, okay? We're going to talk about the other ones in a second. So the first three, they're all going to have DNA, they're all going to have a cell membrane, they're all going to have ribosomes, and then lastly, they're all going to have that liquid, that cytoplasm, all right? So there's your similarities between your prokaryotic and eukaryotic cell. We talked about the ribosomes on here, guys, and making a protein. That got exported from the cell. There's also going to be smaller ribosomes throughout the whole thing here. Those are your free ribosomes, and they're going to make the proteins that are going to stay inside the cell. So these ribosomes make the proteins that the cell will keep and the cell will use. All right. So that's our animal cell. Our last one is going to be our plant cell. I'm going to try to fit it in here on the side. That'll be all right. Okay, so we're going to have the cell membrane. Once again, I'm just going to attach this label to that. We're going to have the cell membrane, but just like the prokaryotic cell over there, the prokaryotic cell had a cell wall, and that cell wall is going to be outside of our cell membrane. Okay, so there is your cell wall. Okay. So similarity again between prokaryotic cells and the plant cell is that they're both going to have a cell wall. All right, so this is your plant cell. All right. They're still going to have a nucleus, just like your animal cell did. All right. Still going to have a nucleus. Still going to have that DNA in the middle. Okay. I'm not going to label everything with this one, guys, because everything is pretty much labeled on that one. They're still going to have the rough ER. Okay. And that rough ER is still going to have the ribosomes on it. So let's put some ribosomes on our rough ER. Okay. Again, guys, I'm speeding this up just a little bit because I just went through all of the functions of these organelles over there. We're going to have our Golgi. Okay. And again, it's going to serve the same purpose as the Golgi in our animal cell. All right. Now here is a difference. Okay. We're going to have a very large central vacuole, or you guys could just call it a vacuole, either way. But what this does is the plant cell doesn't have a cytoskeleton that goes throughout the whole cell. So I know I said I was going to draw it, boom, boom, going all over the place here. They don't have that. Now they have some of the cytoskeleton still, but it's more in its cell membrane and cell wall. So the central vacuole, which is going to hold water for the plant, is actually going to maintain the structure, that square structure of the plant, okay? So animal cells have a circular structure, plant cells have a square structure, and that square structure is maintained by the central vacuole being filled nicely with water. If the central vacuole is not filled with water, the whole cell is going to collapse in. So the cell wall, the cell membrane is all going to be brought in, okay? So that is a difference between plant and animal cells. Plant cell has a very large vacuole. Animal cells have a very little vacuole, all right? Still going to have a smooth ER to make lipids. Okay, so there's your smooth ER. They are still going to have a mitochondria. Still going to have a mitochondria to make ATP. Okay. And they're still going to have our free ribosomes throughout to make proteins for inside the cell. All right. Here is another difference between our plant cells and our animal cells. The plant cell is going to have a new organelle put a label on this one because we didn't go over this yet. And that is called the chloroplast. And the chloroplast is where photosynthesis happens. So it's the plant being able to take in light and convert that light to sugar. That's photosynthesis. So the plant cell is going to have a chloroplast, the animal cell is not. All right. So there is another difference between plant cells and animal cells. All right. Still going to have our lysosome in there. Okay. And there might be more than one. Okay. And that is pretty much it for the plant cell. So again, guys, we talked about the difference between prokaryotic and eukaryotic cells. The prokaryotic cell and eukaryotic cells are both going to have a cell membrane, DNA, ribosomes, and then cytoplasm. All right. Differences between plant and animal cells. Okay. The plant cell is going to have a very large vacuole, where the animal cells is very small. The plant is going to have a cell wall. No cell wall in the animal. The plant is going to have a chloroplast, and the animal cell is not. Okay, so there's three differences there. And again, guys, don't forget the prokaryotic cell does not have a nucleus, and the eukaryotic cells do. Right? The one more thing I wanted to go over, guys, is why don't these cells get real big? All right, we have to make sure that as they get bigger, we are going to have a bigger volume inside the cell, meaning all of these organelles are going to grow. So if we make the cell yay big, we're going to have a very big rough ER. The big rough ER, okay, is going to give us more production of proteins. So if we're making more proteins, those proteins are going to have to get out of the cell. Well, if there's not enough surface area, meaning if the cell membrane doesn't have enough spots where those proteins can get through, the cell will literally fill up with proteins and explode. We don't want that. So that's why cells like to stay nice and small, so their volume inside doesn't get too big. All the organelles don't get too big. We don't have a lot of production or an overabundance of production of things. So that way, it keeps the cell at a good level. All right. So I think this is everything we went over, guys, with respect to the cell, with respect to the cell theory. If you have any other questions, please let me know.